Our project is titled Investigating Genes Specific to Fighting P. aeruginosa Bacterial Infections and Cystic Fibrosis. Cystic fibrosis, or CF for short, is considered an inherited genetic disease that causes patients with these conditions to suffer from increased bacterial infections in their lungs. Buildup of mucus in these bacterial infections can make it extremely difficult to breathe. Thick mucus is the perfect environment to allow bacteria to grow and is why so many CF patients struggle with bacterial infections. Current treatments for these bacterial infections include antibiotics. In theory, they're a great option because they can kill a wide variety of bacteria. However, they can often lead to antibiotic resistance, making the bacterial infection increasingly difficult to treat. Bacteriophages offer an alternative treatment option. Bacteriophages are viruses that infect bacteria by injecting their genetic material into bacteria to kill them. They have a unique ability to target and combat specific types of bacteria, unlike antibiotics, which kill a broad range of bacteria, including healthy bacteria. This also avoids bacteria becoming resistant and running out of treatment options, as there are an estimated 10 bacteriophages for every bacteria type. This specific targeting ability makes bacteriophage an exciting alternative treatment option for CF. The goal of this research was to identify which genes are essential to fight P. aeruginosa bacterial infections for cystic fibrosis treatment. A CF clinical trial sponsored by the University Hospital in Montpellier, France, and led by Dr. Raphael Chiron, combined 10 phages for the treatment of CF patients. This cocktail of bacteriophages were specific in targeting strains or variations of bacteria P. aeruginosa. Our team then found the genomes of each of these phages and used a genome comparison map to find genes of similarity in at least 8 of the 10 phages. These similar genes might be key to fighting these types of infections. We also went a step further in comparing these functions. Our team was able to find and analyze 3 genes of similarity across at least 8 of the phages. For simplicity, we called them genes A, B, and C. Gene A is seen here on the poster in orange. All of the orange colored genes in this image correspond to the similar genes that we grouped to call gene A. All 10 phages had a variation of gene A, making this gene seem promising as a gene that is specific in targeting P. aeruginosa bacterial infections. Of these genes, many of them were reported to have no known function. However, 3 of these variations of gene A reported functions. They included 5' to 3' exonuclease and putative cell wall associated hydrolase. These are very different protein functions that seem to not have a relationship between them. However, they all seem to play a role in phage survival and its life cycle. Gene B is seen here on the poster in purple. 8 of the 10 phages here have a variation of this gene. Of these 8 genes, almost all of them were hypothetical proteins, and only one has a called function, which is a putative structural protein. This putative protein call means that it is not proven to have a function, but rather is thought to be this function based on the DNA sequence, open reading frame, and similarity to other genes. Without further research, it is impossible for us to know whether this gene does help the phages fight P. aeruginosa, or since it is present in most of the phages in the cocktail, it is a promising gene. Gene C can be seen here on the poster in blue. 8 of the 10 phages have a variation of this gene. Of these 8 genes, 5 of them call hypothetical proteins, but 3 call putative proteins. 2 call for a putative tail fiber protein, and 1 call for a putative RNA methyltransferase. Tail fiber proteins play a role in the virus's ability to attach to their host, while RNA methyltransferase could help with the way the virus interacts with the host cell. These probably both play a role in the way the virus infects and attaches to the host bacterial cell, so this could be an important gene in these phages. Because these genes are similar between many of the phages found, they might be key genes needed in the phages to fight P. aeruginosa bacterial infections in CF patients. 3 genes were found to be similar across at least 8 of the phages used to treat cystic fibrosis. No functions seem to be linked to these genes, but this might be determined if more phages are compared in the future. Overall, this similar genes could be a key factor in what makes a phage prime for fighting these types of infections. Most of these genes were recorded to have no known function, but were very similar in amino acid sequence. Therefore, more wet lab work in identifying the function of these genes would be helpful in piecing together what makes these phages special and targeting P. aeruginosa strains. By identifying these key genes, it may make it easier to screen newly discovered bacteriophage or even already discovered bacteriophage in databases like PhagesDB and NCBI to see if they would be beneficial for treating CF patients.